Okay, very good afternoon. We just started like a minute early, so bear with us. I'm just going to take a couple of moments to set a couple of things up here in Zoom. Okay, it's just coming up to 4:30 UK time. I can see the numbers ratcheting up in our attendee list, which is really good. Could you do me a favour? If you've just joined us, could you say hello or hi in the chat, just so I know that you can hear us and that everything's working fine, the chat's working? Hello? Yep, great. Thank you very much. Hello, hello, hello. Thank you for saying that. It's brilliant. Okay. It normally takes a minute or 2 minutes or so for people to join. So whilst that's happening, could you pop in the chat and just let us know where in the world you're joining from? It's really interesting for us to know where our audience is, where in the world you are, where in the UK you are. I'm currently in Southeast London. Oh, this is fascinating. So great. So someone's from Istanbul, London, Switzerland, somebody from East London, a couple from East London actually, somebody from the Northwest, I guess that's the Northwest of the UK. Very, very interesting. So yesterday we did a webinar and we found that there were some people from Indonesia and Singapore. So that's what, so that's at 9:30 in the morning on a Monday. That's who we get. So here we are later in the afternoon. I thought we might get a few more people in the afternoon from the UK because it's the kind of time when people are leaving school and have a bit more time. And I think that seems to be borne out. So yes, thank you very much for letting us know about that. Okay. A very, very warm welcome to this, the second in our virtual taster lecture series of 2026. My name is Kim. I'm the Outreach and Communications Coordinator here at UCL Biochemical Engineering. First thing I need to tell you is that this session is being recorded. We will share a link to it afterwards. So if you miss any of it, you'll be able to watch it back. That's fine. I'll also send a link to everyone who registered. So those who couldn't make it here, I've had a couple of messages from people who couldn't make it. They will be able to join and watch it later. I will be publishing it on YouTube along with all the other taster lectures. So you'll be able to see all the ones that have happened. You'll be able to watch that back as well. But that'll be coming. It will take a couple of weeks though, because we've got we're closing for Easter in a couple of weeks' time. So it won't be happening straight away, but you can watch this back. Okay, so how is this going to work? Well, first of all, you're here on Zoom. Depending on the device you're watching this on, you may be watching it differently. So it's a phone, you might see things stacked like that. You may not have noticed that you've got a series of buttons, so you can do certain things. So as some people have kindly let us know where they are joining us from, you've got a chat function. If you put anything in the chat, you'll be able to message me and Rana, Dr. Rana Khalife. So if you've got any technical problems, if you can't hear us clearly or the slides aren't showing properly or anything like that, let us know in the chat and we'll see that straight away. We're really looking forward to your questions. We very much welcome them. We had some great questions yesterday. We always have great questions in these sessions. It's an absolute joy to take them. But please use the Q&A function. It's really helpful because sometimes we get thematically connected questions, and it's really useful for Rana to look through the questions as well and see if there's a common thread to them. And sometimes if somebody asks a question, it's easier for me to send a link to something else or possibly message back directly. Rana won't be messaging you at all. It'll be me that's doing all the typing. Rana's going to take care of the talking. So so please use the Q&A function. So what I'm going to do now is I'm going to launch a little poll, and it's going to we'd just really like to know a little bit about you. So this is so I'm going to ask you a couple of questions. First of all, I'm going to ask you what level of study would you be interested in. So this is to find out whether potentially you'd be more interested in doing a degree or an MSc or neither. You know, I'll be here just out of interest. But it's helpful for us to gauge our audience whether you're currently doing a degree or currently potentially doing your A levels. So looking at the split that we've got here so far, it's roughly it's about half, just about half to 2/3 people are considering doing a degree, and then some interested in doing a postgraduate, which is really helpful because Rana is actually the admissions tutor for the MSc in Manufacturing and Commercialization of Stem Cell Gene Therapy, as well as being the chair of our recruitment committee. So, you're in very good hands there. So that's really helpful. Thank you for that. The other thing that we'd like to know is whether you have an offer to study with us. It's just to know whether, because we when we promote these these sessions, it's really good to know whether you've gotten off with whether you're somebody who found us because we contacted you through the system, or whether you've gotten off to study, or whether not at all. So this is really interesting because at the moment, the vast majority of people don't have an offer to study with us. Very helpful, very useful to know. So maybe you're new to UCL, new to the department. I hope you enjoy this introduction. So that's really helpful. Thank you for filling that in. That's very, very helpful indeed. Okay, thanks very much for that. So what I'm going to do is I'm going to hand over to Rana, who is going to give the talk, give a presentation on how to translate gene therapy and stem cell discovery to the bedside. I'll be here in the background. I'll pop up a couple of things in the chat. There'll be a couple of links and a bit more information. If you have any questions, get in touch with me through the chat. But I'm going to hand you over to Rana, and then after Rana's finished, we'll launch the Q&A. Over to you, Rana.
Thank you, Kim, for the introduction. So my name is Rana Khalife. I'm an associate professor teaching in the Department of Biochemical Engineering. I'm more into cell and gene therapy. That's why, and I'm biased to cell and gene therapy. That's why I'm giving you this talk. As Kim mentioned, so I'm the regenerative medicine minor coordinator within the Faculty of Engineering. I'm the chair of recruitment in the department. That's why you're going to see a bit of introduction about our department and the degree that we offer. And if I saw 30%, around 30% wanted an MSc. So I'm the program director of one of the MSc program that we have in the department, which is MSc Commercialization and Manufacturing of Stem Cell and Gene Therapy, which is heavily led by industry, where we host, where they host research projects, site visit, etc. And we have people from industry to material. So first of all, can you see my screen, my presentations? My presentation, is that okay?
You just let us know in the chat. That's great, thank you.
Yeah.
And also,
Oh,
just there's
perfect.
one point that I forgot to make. Rana may go into some technical detail here, or some deep scientific concepts. We've had people worry about this in our lecture before. If you join us and do a degree with us, we will take you to the level where you understand that. So I just want to make that clear in case some people say I don't know this, I haven't done this already. We will teach this to you, so please don't worry about that. Thank you very much.
Yeah. So to start with, because I saw someone who said they're interested in biochemistry. I'm a biochemist by training, but I did my PhD in the Department of Biochemical Engineering. And maybe that's why I chose this kind of like topic, because my perspective of seeing things as biochemistry or a biologist has quietly changed when I did my PhD in this department, because this department is more about translational, how you can take the discovery and then translate it into a real life. Okay, so before we start talking about cell and gene therapy, UCL, we've celebrated 200 years of UCL, and we're really known of disruptive thinking. We were the first to in England to welcome women in education, and even people from different ethnic minority as well. And maybe this kind of interdisciplinary thinking, we incorporate this in our department. So of course, UCL is the ninth in the world, as well as fourth in UK and Europe. And as biochemical engineering, we are quite leading when it comes to research in our field. Okay, so we're based in the main campus. So for those who know London, we are near Euston. We have the UCL hospital as well near us. But a few years ago, we've launched another campus, which is UCL East in Stratford, where some of the program, like my program of the MSc, is based there. It's quite really good to have two campuses with a new facility, and with a new campus as well. We have kind of like MFL, Manufacturing Future Labs, where again, we incorporate this interdisciplinary thinking, where we collaborate with different departments for certain tasks in manufacturing. So just to start with our department, our mission is basically to educate the future leaders and then to solve global health. And you're going to see, I'm going to show you, although we're a small department, we have a lot of academics and we have a lot of variety when it comes to research. So although today I'm just going to talk about more cell and gene, but as Kim mentioned, we already have different taster days before. So our strategy is basically to integrate research and education, and this is what we do. Most of our lectures, when we do our lectures, we incorporate real life scenarios that is already happening in industry. We usually invite people from the industry to come and give the lectures to the students, to mentor them, to give them career talk, and even to supervise them and mentor them as well. So we try to incorporate these kind of research education in undergrad, MSc, PhD, and we offer as well other kind of like courses as MBI. Okay, so the BEng programs are accredited as well as the MSc that we have as well. Okay, so what's biochemical engineering? Any thought about what is biochemical engineering? You can pop that into the chat. Um, I have a lot of students asking me, especially in the open days, what's the difference between chemical, biochemical engineer and biomedical and biochemical engineer? We're quite different. So we are interdisciplinary subject where we combine all of the biology aspect, all of the engineering aspect, and we really care about is more about the biology aspect. So maybe in chemical engineer, they care about the process that we care about as well, but we incorporate bio things like alive things. Okay. And this kind of like idea did not come all of the sudden. So it was, so do you know who discovered penicillin? You can pop that in the chat. Yes, Fleming. So he discovered that by mistake, right? And it was smaller scale. But who actually incorporated the chemical thinking or the biochemical engineering thinking was Margaret. And she was able, instead of growing that at a smaller scale, to start think about, okay, how can I produce more? And for us, it's the same thing. So this is what I tell you, like we have a lot of academics that are leaders in their field. We have a lot of research. We have like a vaccine hub. We have two vaccine hub. We have the plastic hub. We used to have the manufacturing of future hubs as well. So although we're a small department, well, not a small, but our cohort size is increasing, but we kind of like have different kind of research field. So for example, for me, I'm more into cell and gene therapy. Even within the cell and gene therapy, I work with pluripotent stem cells. Someone else work with adult stem cells. Or now I'm going to talk a little bit about CAR T therapy or CAR M therapy. We have exosome as well. But we have a lot of people who are more into synbio, vaccine, for example, plastic degradation. We have organoid, we have cultured meat as well, algae, biofuels, and then we have even digitalization and AI tool decision tools and digital twins as well. And then we have a lot of people who are working on the economics because you're going to see it's not only about translational things, is how can you reduce the cost of it to make it available for everyone. Okay, so for us, we take this kind of like idea, and then we need to think about bioprocessing. I'm going to show you why bioprocessing is important and the need that the industry need per year. Okay, so how can we produce this at a larger scale? And I don't know if you can see over here is like a tank. This kind of like a tank, we call it a bioreactor. So instead of growing my cells in a petri dish, or let's say it's T25 flask, which is 25 centimeter square, how I can take this and then even culture it in this kind of bioreactor, which can go up to 10,000 liter or even more. And this is how we teach our students is when you have the discovery, okay, how can you translate this? You don't go, when you have a discovery, if I discover how to treat cancer, I don't go directly and inject that into the patient, okay? I have to have different kind of like steps. So I have to go for preclinical trial and then clinical trial 1, 2, and 3. And the difference between them is the number of patients. Not only this, we teach our students as well is how can you overcome the regulation, for example. So which kind of regulatory body I need, how many testing I need to do before we give it to the patient, and how much is the cost of it as well. Okay, so I'm going to talk about bioprocessing. So when it comes to bioprocessing for us, let's say I'm making insulin from a bacteria. Okay, As a biology, I will take a bacteria, I will reprogram it to produce, I'll genetically modify it to produce insulin. I will grow it, and then I will lyse the cell, for example. I will take my protein, purify it, and then give it to the patient. When it comes to bioprocessing, we have two terms. We have something called upstream, and then we have something called downstream. So upstream is with what is the scale that I'm going to grow my bacteria? Because if I'm a company and I'm producing a lot, I'm not gonna grow it in a petri dish. So we start with the upstream. That means I'm gonna grow it in a bioreactor, starting from 2 liters to up to more than 1,000 liter. And then once my bacteria, for example, produce insulin, I will go for the downstream process. So downstream process are the processes where you're gonna obtain your product. So I'm gonna, for example, lyse my cell, take my protein, purify it, and then packages. Okay. So how do we teach cell and gene therapy in our department? It can range from the undergrad to post experience. As undergrad, if you are biochemical engineering, as BEng or MEng, in your second year, you can take the Regen Med minor. If you're interested more in the cell and gene therapy aspect in the MSc, we have the MSc commercialization and manufacturing of stem cell and gene therapy. You can always do PhD or MBI courses as well. So we're here to talk about cell and gene therapy. So let's talk about RegenMed. RegenMed, and I'm gonna show you 3 case study. But in this kind of 3 case study, I'm gonna talk about more about how can I translate this into a real life and make it in real life. When it comes to RegenMed, is basically we are replacing. So most of the diseases that we are treating, we are currently treating the symptoms. We are decreasing the symptoms rather than treating the disease itself. When it comes to Regen Med, we are aiming to replace or generate a tissue, a cells to establish the function. So basically, we are treating the disease itself. Okay? If you think about it, ages ago, we thought about it like it's gonna be more kind of like a science fiction if we do Regen Med. But then 1997 came, and then they cloned the sheep dolly. And nowadays, like we have organoid on a chip and something like that. And we have a lot of clinical trials happening and some product in market. So Regen Med is not science fiction anymore. So I'm going to talk about tissue engineering. I'm going to talk about stem cells, and I'm going to talk about CAR T therapy. Are you familiar with CAR T therapy? Want to see how many people are familiar with that? Oh, yes. Okay. No, it's all right. That's why you're here. We're going to briefly talk about it. It's emergent of a new cancer therapy. So first of all, tissue engineering. It has been there for more than 20 years. And in this kind of seminar, I want you to think about the challenges. And this is how I teach my students, is we have all of the discovery there. We can know how to make cartilage, skin, bone, heart, liver, everything. This is a discovery. It has been there for 20 years, and still we are waiting for this kind of translational aspect. And this is part of the challenges that we are facing. So let's say tissue engineering. So for example, someone uh, broke their bone, okay? And we want to do it as tissue engineering aspect. Why? Because we have organ shortage, for example, although more people are aware of it, but we're still having organ shortage. So what do? Is we take cells, it could be from the patient, bone cells, or from someone else. We can grow them on a scaffold because the scaffold of the bone is very rigid. We grow them in the lab and then we give it back to the patient. As simple as that, but the reality, it's not. Okay. So there has been some successful kind of like a clinical trial, which I will show you in a bit. Some of them, for example, when we do the trachea that I will show you. So for example, this was the first patient who have received trachea. So this patient have her trachea collapsed. So what they did is they took the trachea of a pig because it resembles, it's similar structure as a human. They remove all of these cells from the animal and then put the patient's cells on it, grow it in the lab, and give it to her. And it was very successful. Now, it's a good thing that we now can treat it. We don't need like organ donation and so on. If I show you how it's done, you can see the trachea, like it's like whitish. When we usually put the cells, it will become red. And then I don't know if you can see on the right-hand side, it's quite kind of like you put it on a roller bottle and then you grow it. Okay? If I'm manufacturing, this is not quite feasible. There's a lot of challenges here. So for us, we teach the students, okay, how can I take this? How can I design a bioreactor when I put the scaffold, for example, and I put the cells and I put all of the nutrients, all of the cells will be equally distributed throughout the scaffold. They will receive the same nutritional factors and so on. And how can I make my process robust so I can produce it at a larger scale? Okay, so these are some of the challenges that we are facing. Now, stem cells. Okay, so I'm going to talk about pluripotent stem cells. So pluripotent stem cell can give rise to many different types of cells in your body. Okay, we don't have pluripotent stem cells in our body. We have adult stem cells. Adult stem cells like hematopoietic stem cells that give blood cells. We have mesenchymal stem cells as well. So when it comes to pluripotent stem cells, yeah, we in 1998, Thomson derived the first human embryonic stem cells, and everyone was rushing towards to do clinical trials and so on. But then it was stopped. Why do you think it was stopped? Ethical debate. Okay, until in 2006, 2007, where Professor Shinya Yamanaka, the Nobel Prize winner, Japanese Nobel Prize winner, in 2006 discovered that you can take any cells in your body, reprogram it to become pluripotent stem cells. By doing this, there's no ethical debate. If someone, for example, lost their vision, we can take skin cell, reprogram it to become pluripotent stem cells, and then differentiate this into photoreceptors and then transplant it to the patient. That was a revolution, and we're celebrating more than 20 years of iPSCs. And it's kind of become autologous therapy. So autologous therapy, that means it's from the same patient to the same patient. So the immune system is not going to recognize it. Amazing, right? 20 years. Do we have a lot of clinical trials? Yes, we do have a lot of clinical trials when it comes to pluripotent stem cells. We have clinical trials to treat AMD, so people who have lost their vision, um, diabetes, and other kind of like clinical trial. Do we have a product in market? No. Another challenge, okay? How can I translate this? So if you want to know how we can translate this, you need to take one of our degrees, undergrad or postgrad. So for us, this is how we teach the students. How can we overcome the challenges? We can apply, for example, this is from my own data from the paper because I was working on pluripotent stem cell, is by applying engineering concept to optimize, for example, the differentiation. Because, for example, the cost of these kind of therapies are a lot. So for me, what if I can optimize the differentiation? Maybe I can lower the cost. That means and I can make, of course, the process more robust. Okay, so I know that I'm going for three kind of case study, but the three of them have challenges, and these kind of challenges, this is how we try to overcome the challenges. Okay, now I'm not sure if this will work, but please let me know if you can hear this video. If not, I can speak. Can you hear it?
I'm not. No, I'm afraid we're not getting any audio.
Okay, so this is, I will speak while you can see the slides. Okay, so this is Emma. Emma was the first in the world to receive this kind of CAR T therapy. people on this clinical trial is that sorry, I have the voice here. Um, they first started treating these people who had leukemia. Okay, it was their last hope because nothing has worked, none chemotherapy, nothing. So what they do actually is, I'm gonna show you. I'm gonna jump into this. So, who's responsible to fight cancer in our body? It's like T cells, your immune cells. So, what they did is they took the T cells of the patient, they genetically modify it to express on its surface something we call a CAR. Okay. This kind of like receptor, when we give it back to the patient, it will go and target only cancer. And it was very successful. So Emma, it was her last hope to get this kind of therapy. She got it in 2012, and till now she's cancer free. The She did not relapse. The cancer did not come back. The only problem is that they had to put her into a coma. Okay, I don't know if you know. So when you have T cells or you have inflammation and so on. Okay, sorry, I was just reading something. So you already know that when you have usually inflammation and so on, it's like it becomes like red, or like you feel like there is a flush of blood and so on. So when they did give back her T cells, there was something called cytokine release. It's because of all of the things that the T cells has produced. So they had to put her into a coma. Now, we started with two products in market to treat blood leukemia, but nowadays we have more than 10 approved product in market, and we're starting to cure as well solid tumor. So what I'm showing you here is Emma, and then even like she started her own foundation to go and raise awareness when it comes to what is CAR-T, because there is a lot of ethical debate. People will be like, no, I don't want you to take something from me and then genetically modify it and give it back to me. And this is the picture of Emma with President Barack Obama, and just to show you her character. She was like, the president was like, what can I do for you? You can ask me anything. So if a president asks you for anything, what would you ask for? Maybe money? She was like, my school don't know that I'm here with you, so please can you write me a letter to excuse me from not attending the school? This is how dedicated she was. Anyway, let's talk about UK. Where do we stand for? We have Layla over here, and she was the first UK patient to receive this CAR-T therapy in 2015. And we're really proud as UCL, sorry, as UCL to have a spin-out company from us called Autolus. And then they got their first CAR-T UK approved market not last October, October 2024, which is a big thing for the UK. And they're on BBC, so you can watch their story as well. Okay, it's good. What could go wrong with this? Any thought? We're curing cancer. What could be the challenge in here? Make other blood? Oh, interesting. Good mention. Mass production is fine. We can do so. Most of these therapy are autologous. Autologous, as I mentioned, is like from the same patient. We're trying to make it allogenic, so from one donor to many. When we make it allogenic, that means we can, someone said mass production, which is good. We can make it bigger, then we'll reduce the cost because someone really got it right. It's the cost. So, how much does it cost? Any thought? Eh, fair enough. So, when, as I mentioned, we used to have two product in market, but now we have more than 10 product in market. The first one released, we have Yescarta and Riscarta and Kimria. It's around 500,000. Okay, so at the beginning, the NHS did not provide that, but then they negotiated with Novartis to sell them at a lower price, and then some people from the NHS, well, some patients are receiving it. Okay, so the main challenges in CAR-T. other than the cytokine release is basically the cost. How can we make it allogeneic? By making it allogeneic, can we grow it in a bioreactor, reduce the cost as well? It's $475,000. Okay. Someone just asked me. Okay. So UCL is leading when it comes to clinical trial, and we have a lot of clinical trial when it comes to cell therapy. So what I want to show you is basically we're taking part not only at, well, translating things to the last thing to the market, but even in the clinical trials. And then maybe after the clinical trials, how can we make things at a larger scale? It's not as easy as you think about it. And I like this diagram as part of our undergraduate teaching in my minor, is basically when you manufacture things at a smaller molecule, it's really easy. The bigger it gets, like you can see part of the cell here, the harder it gets, and you'll have more complexity and more challenges. Okay. Now, the importance of having the background of bioprocessing, and this is what I tell my students even in the open day, is highlighted by the FDA commissioner, Scott. He's like, we know the discovery, but we don't really have people with this kind of like knowledge to translate or like how to manufacture things at a larger scale. So this is really needed. And I was in a meeting when it's kind of like between academia and industry hosted by Catapult, and they were like, before the pandemic we wanted around like 400 people with bioprocessing background each year, but after the pandemic this has increased, and we need to secure like around 8,000 jobs for people who have bioprocessing background, because this field is increasing dramatically. That's why we need people with this kind of background. It's not easy. So we've mentioned Kimbria, but I did not talk about gene therapy, but other gene therapy products are expensive and it can go up to 1 million like Gliberia. Okay. What's our role? Basically, what is our manufacturing scale? How can we grow the cells in these kind of manufacturing scale? You have cells that are adherent, so they need to adhere on something. So we cannot directly grow them in this bioreactor. We have suspended cells. Okay, now applying engineering concept when the cells are in this kind of like bioreactor, what is the spinner speed, for example? Will the shear stress affect the cells and so on? So I think that's it for me. So we need to think about the whole bioprocessing approach when it comes to when we collaborate with company to optimize the processing, we look into each kind of like step in the processing. For example, how can we optimize it and what's going to be the scale of it? Okay. I mean, that's it for me. I'm happy to answer any question.
We've had a flurry of questions. I just wanted to just go back a little bit and say that some of the comments we got when Ron was asking the questions were really good. Some people said that the risks of this treatment, things like people said cost, production, the risk of attacking healthy cells, and somebody mentioned autoimmune conditions and cytokine storms, which is, this is very very interesting that you're picking up on these.
Okay.
And the estimates that you had were between sort of 30,000 and a million. And I'm not sure which currency that was. Somebody said $1 million. So yeah,
Dollars.
whether you're talking pounds or dollars, I mean, it's the number of zeros at the end of it that's really important, I think. What I'm going to do is because we've already got
Question. I
questions that
will
have
go
come in.
for it.
So I'm going to let Rana just have a quick scan through those, have a read through them, and while
Yeah.
Rana's doing that, I just want to shamelessly plug that we've got some more taster lectures coming up. So let me pull them up in front of me. And I will just mention that we've also got one coming up tomorrow, which is going to how we can accelerate vaccine manufacture without using cells. And the real advantage of this is that we don't have to use cells. We don't have to keep the cells alive in the same way. It could be, you know, how do we use this new technology to potentially speed things up, make it cheaper, and use far less energy and resources? We're also doing one, which is I'm really looking forward to this one on the 27th, which is how soon before lab grown meat just becomes meat, and we don't actually think of them as being any different. The last one is going to be Matthew Banner, and he's talking about how the biopharma sector is using AI and automation. And the session that we did yesterday, a lot of people were asking about artificial intelligence, how it can be used, what difference it's going to make. So come along to Matthew's session. If you can't make it, just register anyway. I'm going to pop a link in the chat. So yes, somebody's just asked you to be linked to the or email it to the chat. Yes, if you've registered for this or any of these webinars, I'll send a link out in the coming weeks. It'll take a while to get them all uploaded, but I'll send a link to all of the recordings so you can watch any of these back in the series. They'll all be available to view back. So I'll just create a page with all the YouTube videos. If you can't watch YouTube where you are, there'll be a Zoom link as well, so you can watch back the Zoom recording. But now that I can see that we're getting more questions coming in as well. Just to let you know, Rana, there's one that's coming through the chat as well. If you could use the
Okay.
Q&A for the questions, it's really helpful because we can sort of tick them off as we go along, so we're less likely to miss one. But I'll pop a link to the rest of the sessions in the chat now and hand over to Rana.
Yes, thank you, Kim. Okay, so when there's a question, I'm gonna read it loud. When you use pig's trachea to treat the patient with collapsed trachea, is it correct to say that the one part left of the pig is the scaffold or the bone? It's just like we call it extracellular matrix. It's just the how it looks like, but there is no animal component, and it shouldn't have any animal component. And it's part of the quality control testing that we do is we need to make sure that there is no animal cells or animal component howsoever. Maybe the collagen, maybe the extracellular matrix, but not the cells. Okay. What are mesenchymal stem cells? Mesenchymal stem cells are adult stem cells that can give rise to specific kind of cells. We can find them easily in the fat tissue, for example. When we differentiate these MSC, we can differentiate them into neuron, we can differentiate them into bone cells or chondrocyte as well. So we have them in our body. These are adult stem cells. Is it possible to use the Yamanaka research for healthy cells to remove mutation and reset their age for egg? Interesting. I mean, you need to consider something that when this is more gene therapy rather than cell therapy. Gene therapy is when we use CRISPR, for example, or viruses, lenti, and so on, to correct a mutation. Now, if I correct a mutation in my cells, this cell is not going to regenerate. I'm going to have the precursor of the cells to regenerate my cells, and my cells will die. So if it's, for example, in the initial egg and I'm correcting this kind of mutation, then yes. But if it's already a somatic cell that is already differentiated, then no. Is leukemia treatment publicly available or it's still in clinical trial? So no, when these products get the approval that and you have a price tag for it, that means it's already in market. So we have a lot of more than 10 CAR T in already in market. You mentioned the application of CAR T therapy in solid tumor. What are the challenges in applying it to solid tumor rather than hematological malignancy? Very good answer. Very good question. Okay, so when it comes to leukemia, it's in the blood. The blood is circulated throughout the body, right? Now, when it comes to solid tumor, first of all, and usually when we give the CAR T, it's gonna be in the vein, okay? Now, when it comes to solid tumor, first challenge is how you're gonna inject that. Are you gonna inject it in the blood? Then, how are you gonna make sure that the T cells that are injected in the blood will go to the side of the tumor? Not only this, when it comes to solid tumor, it depends on the stages. As you know, like the solid tumor will have kind of like 3D aggregate, okay? The bigger it gets, we have kind of what we call a hypoxic condition, which is like low oxygen. Now, we need to make sure that these T cell will resist this kind of like condition. So I'm not going to go more into details, more complexity of it, but yeah, this is the two challenges. And it will be like at which stage we're going to give this.
Yeah, and just to say, one of the issues with a tumor, especially something like a brain tumor, as Ron was saying, as it grows, there is no blood getting to the center of it, so you can't actually get a lot of therapies
Mm.
to it. There are certain things that can improve this. There are certain therapies that can improve it, but it's really challenging. It's why brain tumors are very, very difficult. One of the reasons why brain tumors are so difficult to treat. I hope that as many of you as possible don't come into contact with this for as long as possible. But if you do consider it as a professional challenge, please do. It's an area that really, really needs support. But the challenge is, cancers are very different depending on where they're in the body and the kind of mutation. Because when your cells mutate uncontrollably, they can do so in many different ways depending where they are, the kind of cells that it happens. They can be treatable in different ways. It is really difficult, and it's only like in the last decade, 2 decades that we've had really good treatments for them. So this is why it's so exciting, but also still really challenging. Just also, I just want to say, Rana, there's somebody's put some really good questions in the chat. If you could put the questions in the Q&A as well, it's
Yes,
really helpful
please.
for us to read them because they just disappear in the chat for us, you see, so it's harder to keep track of them.
Okay, does CAR-T have advantage for patient where chemotherapy does work? So let me tell you something that for now, CAR-T therapy is offered for patients who have like they've already went through chemotherapy and all of the therapies and immunotherapy and nothing has worked. So it's the last line kind of like a defense for now. Hopefully, we'll try to convince the healthcare to put it as first. Okay, I'm sorry guys, I'm just taking. Okay, so practicing. What's the main difference between biomedical science, biomedical engineer? I cannot answer this fully because I'm not aware about the curriculum of the biomedical sciences, but I can tell you the difference between them maybe in general and us. As I mentioned, we care about the bioprocessing with incorporating living things as well. Is it possible common for patient body to reject the pig's trachea? Yes, if there is a contaminant from the animal. So yeah, maybe sometimes we have some fails in the clinical trial. Well, when we transplanted the trachea, it collapsed all of the sudden, and we don't know why. Does metastasis make it hard for cell therapy to actually work? Uh, I mean, when it comes to CAR-T therapy, we can always argue that it's considered between cell and gene therapy because it's considered cell because we're giving the patient a cell, a T cell, and then gene therapy because we're genetically modifying these T cells. Um, that's why it's another challenge when it comes to solid tumor and metastasis. And at which stage we're giving the CAR-T? Is it early stage before the metastasis or after that?
Sorry, I just want to take a moment and say somebody was asking about biomedical engineering. What I've done is I put a link in the chat for the degrees that biomedical engineering does at UCL. So it may be really helpful. It's always really good to have a look at their site. You can see the modules that compose it, but you'll see that a lot of a lot of their degree courses around the actual physical interaction of technology with the body. So a lot of biomedical engineering, sort of medical physics, is things like MRI scanners or radiotherapy for cancer, medical implants. There is a certain crossover with biochemical engineering. Our own Professor Chris Mason, he's as much a biomedical engineer as a biochemical engineer. So there's a lot of crossover between the subjects. But do have a look at that link if you want to know more about biomedical engineering, and you can see the difference with our degrees and what goes to comprise it.
I
Yeah.
just have someone on the. So is it likely to get into biochemical engineering degree without doing biology A
Yes.
level? Yes. Yeah.
Yeah. Absolutely.
Um, in everything, like even in my minor is regenerative medicine, across the faculty. So I have, for example, people from computer science, and then I have people from chemical engineering that have nothing, like no background when it comes to biology. And this is how we do it over here. Like when we first start the module or the course or so on, we build up the knowledge. So we start with the basic of biology and chemistry, even basic of physics, for example, if you don't have any physics, and the material will start to build up. I'm not sure about one of the questions, but I can answer the second part of it, which is why can we make this therapy affordable on the ground and developing countries? I mean, this is part of what we're doing as like we know each, for example, with CAR T, we have to take T cells, we have to activate it, we have to use lentivirus as well to genetically modify it, expand it, and then so on. So we know each step. So we try to optimize it to try to see because the material, sometimes the materials or the growth factors that we use in the process cost a lot. So for example, to differentiate stem cells to liver cells, I have to use one of the growth factor called activin A. Activin A for 1 ml or even less costs more than 1,000 pound. The media of the, so usually when you put nutrients on the cells, 500 ml bottle for normal cells costs $10. For stem cells, 150 pound. Okay, so this the process and the raw materials that we use are very expensive. So once we try to optimize this, then yes, we can make it affordable to all countries as well.
Okay. Whilst we please carry on your questions. We've kept until the end of the half hour for for these questions. But you know, if we answer them all, we're happy to end the session. But before we do that, and to give you a chance to ask more questions, I just want to say that you asked people have asked things about, you know, can I do this course about chemistry? Can I do this with this talk course about biology? Or you know, or some people come without chemistry and biology. They have physics. They find some things easier than the people who didn't do physics. Some people come with biology but not chemistry find some things easier. Likewise, what we find from the response from all the students that have spoken to me about this, they say that whatever you come in with, you have to learn something else, and there's always more to learn. Our modules don't assume that you've done biology or chemistry or physics. You have to have done one of them. As for the B engineer eng, you have to have done maths as well, not for the BSc, but you need to have done some kind of science. But you know, our modules are designed to take you up to the level. We also assume that if you're you're going to be self-starting and motivated to learn because you're going to university. Okay, somebody's saying that they're having an issue accessing one of the links. I'm going to go and sort that out. But I'm also going to hand over to Rana to talk about the in-person open days coming up at the 26th, 27th of June,
Yes.
Friday and
I mean,
Saturday at the end of June.
if you would like to know more about our undergraduate degree, feel free to join the open day. So we're gonna be there in Robert's Foyer, if I'm not mistaken. You can ask us directly questions, and we're gonna have lab tours, and we're gonna have as well kind of like a session where our UG admin we'll talk about more about the criteria and the degree, and then basically which kind of jobs our undergrad go for. When it comes to bioreactor, I saw just one question. So um, of course, all of these bioreactors that we use, we have to test them. Okay, so to make sure, for example, to know how much nutrients to put, we kind of study the cell metabolism, for example, to know, for example, how much the cell needs like glucose or nutrients, the consumption rate of the cells, the oxygen as well. So once we do this, for example, with CAR-T, the expansion is in wave bags, so it's not in kind of other kind of like bioreactor. But what we have done with Professor Kasim Rafiq is we try to optimize this to grow in one of the type of the bioreactor, and it was one of its kind. So this is part of our research. Is we take these cells and we try to know how can we grow them in bioreactor as well. Um, I'm not sure if there is more questions.
I think that's it. So I put links to the next three webinars. The next three webinars are how we can accelerate vaccine manufacturing without cells, how soon before lab-grown meat becomes just meat, and how the biopharma sector is using AI and automation. So those are in the chat. Just want to say when this webinar finishes, obviously the chat's going to disappear. So if you just go to UCL Biochemical Engineering's page, you'll find all of the events on there. I've also put my email address in the chat as well. If you have any questions about studying at UCL, if you're interested in maybe the MSc by manufacturing commercialization of stem cell and gene therapies, or a degree, or you want to ask a question which you didn't get a chance to ask now, please do get in touch with me. It's very easy to find my details through the website. I'll be happy to forward you on to somebody else. Otherwise, just once again to say that this session's been recorded. There will be links sent out afterwards. It will take a couple of weeks because we've got Easter coming up as well, so we're going to be closed for a couple of weeks for that. So, but we will get links to all the recordings. You can sign up to any of them, but it'd be great to see you tomorrow, Friday, and Monday for those. It'd be even greater if you could join us in person at some point at the end of June, and we can show you our laboratories, and you can meet Rana and myself. But in the meantime, thank you very, very much for recording. I hope you have a great rest of the week. I wish you all the best with your studies or whatever you're doing. And thank you for your questions. They were really, really engaging. It's great to have them come through. Somebody has asked, and this is an important one, how you sign up for open days. So the tickets aren't available yet, but if you go and sign up if you go to UCL and just search the web page for open days UCL events, you'll be able to register your interest. You can also, what I think actually that's an important point. Just because you've signed up to this webinar doesn't mean that you've signed up to contact from UCL. They're separate things. So I will send a link out included with a link to watch the videos, so you can see the videos, recordings of these. I'll also send out that you can register for updates from UCL. So you'll find out any more lectures that are coming up, taste of things, events, open days, all the kind of stuff that we do here. And it's UCL's 200th anniversary. We're 200 years old as a university this year, so we've got loads of exciting stuff. But I'm going to wrap that up now. Rana's had to jump off to do with students. But no, thank you for your kind comments. And thank you, Rana, for giving an excellent talk. It's been great to have you here. Have a great rest of the week, everybody.
Thanks, everyone.